Dear students, welcome and thank you for tuning in to this video. We are continuing our discussion about the structures inside the body and protein synthesis. In this video, we're going to talk about DNA transcription. Transcription is the first of several steps of DNA-based gene expression, in which the particular segment of the DNA is copied into RNA. And we are doing this in order to protect the DNA strand from damage. It holds our genetic information and should be preserved. That's why it's in the nucleus. The process that we are going to describe, which is the transcription, happen inside the nucleus. So we need to transport the information from inside to the outside of the nucleus. And in order to do this, we use RNA. The players in this process are the RNA, ribonucleic acid, and also the enzymes, which is RNA polymerase. So any term that ends with A's is an enzyme. And enzymes are catalysts that speed up the process of or a reaction, and in this case, speed up the formation of RNA. So inside the cells, we have the nucleus. Nucleus has the DNA. We need to make a copy of the DNA and to move it outside the nucleus. This copy is made through using the RNA. RNA are base pairs of nucleotides as complement languages. So you will have one that oppose the other one in order to make sure that we have the sequence and the gene that is made of the sequence of those nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. And in the DNA, we have actually 4 bases, which are adenine, thymine, guanine, and cytosine. Guanine will get attached with cytosine, and adenine go attached with thymine. And that's what I meant, that they're pairs. They pair with each other. So remember, the DNA is a double helix strand. This is one strand and the other strand, and the information between is the DNA bases that holds the genetic information for creating all the proteins inside the cell or inside the body. The RNA is a sequence of also 4 building, and they are called RNA nucleotides, and they consist of 3 parts as well: a sugar, a phosphate group, and also a nitrogen containing base. The bases in the RNA are adenine, uracil, guanine, and cytosine. If you notice that it's similar to the DNA bases, except that we have uracil instead of thymine. So they're very close in structure. They can make copies and complements of copies to give us the information that we need. So to summarize how transcription is happening, I'm going to provide a quick overview that is relevant to what we are going to use it for in about the biomaterials class. So if you look this schematic, this is our nucleus, and we have our DNA, and this is our DNA strand. And you can see this small box here. It's magnified in this image, and this is a place where the DNA is being transcribed in order to create messenger RNA. So all the small dots are messenger RNAs. I don't want to go into details about these components, but one of the interesting parts of our chromosomes is actually something called telomere, and telomeres are segments of our chromosomes, the end parts, the sides of the chromosomes. And interestingly, there is research that suggests that as the cells divide, this chain is getting shorter, which means that they can serve as a biological clock. If the telomere reaches a certain length, then the cell will commit apoptosis and die. Interestingly, in cancer cells, this telomere length stays constant, so it does not shrink, similar to what happened in normal cells. And there's a lot of research in cancer therapy about this, about what does this mean, and how can we control the length of the sides of the chromosomes. And keep in mind that we said before that in the DNA we have gene sequences, gene strips, and we have non-coding DNA, and telomeres is a non-code. What we think is they're not non-coding DNA, so it does not transcribe and translate into proteins. So it's an interesting thing to keep in mind if you're studying tissue engineering, because this is important. Going back to this small box here, to talk about how we are going to do how transcription is happening. So you remember from the previous video, this is a chromatin, which is a collection of a lot of nucleosomes. Each nucleosome is made of 8. We discussed this in the previous video. So these nucleosomes are made of 8 histone molecules, and the DNA is wrapped around them. So this is what we are seeing here as well. So those are the nucleosomes. This is our DNA. Those are the histones. And then you have this strand of DNA. For a cell to start creating those RNAs, we need to have a signal that comes to the nucleus to start a sequence to do something called upregulation of the gene. So see these nuclear pores. Some proteins will have to get inside the nucleus, and then they trigger the sequence that we are going to, let's say, if this gene is for insulin and we want to create insulin, an upregulation signal, a protein should come in and start this sequence there where we need to upregulate the insulin gene. So what happens if this signal comes in that we want insulin to be produced? We need to go to the sequence that holds the genetic information of insulin, and then something called RNA polymerase, this is another protein, should come into the DNA strand and start creating something we call messenger RNA. So the messenger RNA, from its name, it's messenger because it's going to transport the signal from inside to outside, and it's an RNA, which means that it has adenine, uracil, guanine, and cytosine. So this is the strand here. This is the pink strand, and same thing. We're going to use, the body is going to use histone molecules to create this messenger RNA, and then we have after the complete gene have been transcribed, we have our messenger RNA, and this messenger RNA need to go outside through the nuclear pore to outside the nucleus to start something we call protein translation, right? So this is a transcription, RNA transcription, and then we have to translate the messenger RNA into proteins. This will provide a picture about what's happening inside the nucleus, because this is very important from a biomedical engineering point of view. If we need a certain protein to be produced, because this is what we do in biomedical engineering, we need to control the fate of the cell. And so we needed to create proteins, specific proteins. So we need to stimulate the nucleus with the signal that is going to upregulate or downregulate. If we want to stop the production of protein, we can downregulate. So these signals are the things that the biological molecules or proteins that we might be seeding inside our scaffold to control the fate. After you get the signal inside for upregulation, this chain of sequence of reactions will start by the RNA polymerase, the opening of the DNA strand, and creating this messenger RNA to go outside of the nucleus. Thank you for watching. Until the next video.